Hello students, welcome to this new lesson of cell biology in the Cell Biology School. Today, the DNA molecule. So everybody knows that DNA is present in every cell and it is usually located in the nucleus for eukaryotic cells. However, in prokaryotic cells, there is no nucleus per se. There is still a molecule of DNA which is free-floating in the cytoplasm. So the DNA molecule is a double helix. It is made on a sugar phosphate backbone, meaning that all the nucleotides making the DNA are linked side by side by their sugars and phosphates. And there are two strands of nucleotides. Now, in the middle, the two strands are linked by the means of hydrogen bonds linking the complementary nucleotides. The molecule twists due to its structure, forming two grooves alternating, a minor groove which is the smallest and a larger groove which is called the major groove. So if we look more precisely at the nucleotides entering in the composition of the double helix, first of all, there are four bases or four different nucleotides in the DNA. Two of them are pyrimidines, it is the C and the T, and two of them are purines, the A and G. Purines can only interact with pyrimidines, and pyrimidine can only interact with purines. More than this, the T's can only interact with the A's and the C's can only interact with the G's. So if we look closely at the bonds making the DNA double strand, we have first linkages between side nucleotides or neighbor nucleotides. These bonds are phosphodiester bonds and they link tightly the neighbor nucleotides in the same strand. They are very unlikely to break just because these bonds are very strong. In the middle, so between the two facing strands, it is hydrogen interactions. Each one of these interactions is relatively weak, but all together they make a relative force. And over the length of the DNA, with many nucleotides, each of them forming either 3 or 2 bonds between A and T's, it makes a double helix with two strands that are pretty well attached and held together due to the numerous hydrogen bonds in the middle. It's a little bit like a zipper. Of course, the more you have C and G's pairs, then it will be more difficult to separate the two strands. In contrast, where there are lots of T and A's or T and A pairs, it is easier to separate the strand because you have less hydrogen interactions. This is why in areas where transcription starts or where replication starts, there are lots of TA pairs because it's a bit easier to open than the CG pairs. This is it, folks. Come back later for a new cell biology lesson.